Who's playing some music in the background?
Oh,
Oh, was that you? All right, let's move on to the first of tonight's lectures for Applied Genomics and Plant Breeding. And this lecture will be on genomes and genomics. So, by way of introduction, let's consider an important finding in 1997. But so we'll just go back to this thigh bone that you can see in the image here. This was a thigh bone fossil that was discovered in 1856. And in 1997, some researchers at the University of Munich, they actually took a bone sample from this fossil and they tried to extract some DNA and sequence that DNA. And it was actually quite astounding, the achievement that they were managed to report the sequence of a 379 base pair region, not of nuclear or chromosomal DNA, but from mitochondrial DNA. But they successfully sequenced that region of DNA from this very, very old fossil. And as I said, this was an astounding achievement because, as you can imagine, DNA molecules break down over time. So the amount of DNA present that was actually intact enough to sequence was very, very small, and the scientists had to take a lot of care to make sure that that the sequence was not actually just some contamination from modern day humans. But it was actually very exciting because the sequence actually showed or indicated at least that the Neanderthals became extinct without contributing any mitochondrial DNA to today's modern humans. And sounds like someone's trying to get on. And of course, the Neanderthals were thought to have gone extinct about 40,000 years ago. So 379 base pair region of DNA. 13 years later, the same research team at the Max Planck Institute, very famous institute by the way, obtained 4 billion bases of sequence, of DNA sequence from 3 Neanderthal fossils. So look at the advance in the technology of genomics from only 379 bases to 4 billion bases. It's quite astounding, these dramatic advances in this technology over the last few decades. So let's look at some examples of genomes that have been sequenced. And when we say a genome is sequenced, that means that they actually identify the order of the nucleotides, that is the bases, within right along the length of that particular piece of DNA, whether it's a chromosome or another piece of DNA, which is in one of the organelles. So mitochondrial DNA, chloroplast DNA as examples. All right, so in 1995, the genome of this bacterium, Haemophilus influenzae, was the first genome to be sequenced, the very first genome to be sequenced. The size of that genome was 1.8 megabases, where 1 megabase equals 1 million base pairs. And this bacterium, Haemophilus influenzae, just for your interest, it was actually thought to cause the flu, influenza, right up until 1933, and that was when the flu viruses were actually first characterised. So it was a virus that causes the flu, not bacteria. But of course, this bacterium has retained that name, Haemophilus influenzae. But it does cause blood poisoning called bacteremia. It causes pneumonia, causes bacterial meningitis and various infections, if you're interested. Then, the following year, 1996, the 12 megabase genome of the yeast, Saccharomyces cerevisiae, was reported. In 98, 100 megabase genome of this nematode worm called Caenorhabditis elegans. In the year 2000, an even larger genome, about 180 megabases, of the fruit fly, Drosophila melanogaster. And then the very famous reporting of the human genome in 2001, 3,000 megabases worth of DNA sequence. By the end of 2009, 2,000 bacteria, 88 fungi, 14 plants, 31 mammals had their genomes sequenced, and many others. And currently there are well over 1,000 species that are having their genomes sequenced. So quite amazing, that rapid advance in genome sequencing technology.
Well, is that the entire genome or just sections?
That's the entire genome. The entire genome. Oh, but I should say, if you talk to anyone that's involved in genome sequencing, they will always tell you we can never sequence the entire genome. They will always say genome, the sequencing of the genome is never complete. So you may have heard that from Pippa in some of your previous lectures, that even though with all the technology in sequencing, there's still a lot of grey areas around whether a genome sequence is ever really complete, given current technology. So yeah, good question there. All right, so genomics. Let's think about genomics. Genomics has really revolutionised how genetics and genetic analysis is performed, and it's opened up all these avenues of inquiry that weren't even conceivable just years ago, just decades ago. So far, you've probably, in this subject, you've probably looked at what they call a forward approach to genetics, and that's called forward genetics. And that means that you look for a mutant phenotype in an organism. So some mutant observation in a plant, for example, and then you characterise that mutant to try to identify the gene that's causing that mutant phenotype. And then, of course, you try to characterise the function of the gene, the RNA, and the protein sequences. So that's called forward genetics. In contrast, having the entire DNA sequence of an organism's genome available allows researchers to work in both directions. That is, they can perform forward genetics, and they can also perform what's called reverse genetics. So using reverse genetics, researchers can really study the roles of formerly unknown genes. So with reverse genetics, of course, you would look at the gene itself, start with the gene itself, and then try to work out the function of that gene without even looking at a mutant phenotype. So genome sequencing has revealed many, many new genes, and we can now more easily study the roles of previously unidentified genes. So analyses of genomes now contribute to all these different aspects of research, biological research. And these are just some examples here. So in the area of human genetics, the field of genomics can provide new ways to locate genes, for example, that contribute to genetic disease. In terms of model organisms that have been used to study genetics, particularly long-studied model organisms, the availability of genome sequences has really accelerated the identification of genes, the analysis of gene function, and even characterising those non-coding regions that you find in between genes. The next point here, systems biology. This is a relatively new field that developed, and systems biology really enables the use of new technologies for analysing the roles, physiological roles of all gene products. Genomics has also opened up new avenues of study in evolution and in ecology, and even in the area of human medicine. Has anyone seen that very old movie, 1990, I think it was a 1997 movie called Gattaca? Anyone ever seen that movie? Gattaca?
No.
No?
No.
I think it was G-A-T, Gattaca. If you get a chance to see it, this is really mind-boggling. Yeah, this movie here, Gattaca. It was based on what they call eugenics. That is, they use genetic selection of future children by selecting embryos that are seen to have superior genetics, superior hereditary traits. So when a woman wants to fall pregnant, they would look at the different embryos, and they would say, okay, this embryo, the genetics of this particular embryo has a 50% chance of developing alcoholic traits when they're, adults, or 60% chance of, developing criminal tendencies and so on. So a couple would actually choose the embryo that was thought to have, ideal traits, intelligence and athleticism and so on. So a really confronting movie, particularly when you see or when you understand that the genetics that's available could potentially one day lead to that sort of scenario. So if you get a chance, Gattaca, actually I think it was double T, Gattaca. And this name Gattaca, you might have noticed, includes the 4 nucleotides of DNA, A, T, C and G. Yeah, very interesting. Ethan Hawke was in that movie, Ethan Hawke and Uma Thurman. You might know them. All right, so in this lecture, we're going to focus on 3 major aspects of genomic analysis. First one is bioinformatics, number 2, comparative genomics, and number 3, functional genomics. So bioinformatics, shown here, requires or involves the analysis of the information content of the entire genome. Bioinformatics is normally carried out by individuals that are very, very good with their computing skills, very good. They spend a lot of time on the computer, using computer programs, normally really high power computers. So bioinformatics includes the numbers and types of gene products, as well as the location of those genes, the number and type of binding sites within DNA and RNA that allow functional products to be produced at the correct time in the correct place. So this is generally referred to as temporal time and spatial place, temporal and spatial gene expression. In other words, when is that gene expressed and where is it expressed? All right, next we have comparative genomics. So if you consider that a particular DNA sequence which is conserved among different species of organisms, if it's very highly conserved, that is, it's the same or very, very similar, this provides a clue as to an unknown gene function if that same sequence is found in the DNA of another organism. So, for example, if you've actually listened to last week's lecture, you'll remember that homeobox domain. That's present in homeotic genes, not just in the fruit fly Drosophila, but in many, many other species, including plants. So this field of comparative genomics considers the genomes of closely and distantly related species for the purpose of evolutionary insight, and also allows these conserved sequences to be used as a guide to predicting gene function. If you were, for example, studying the function of a completely unknown gene. All right, and finally we have functional genomics. So this functional genomics includes the use of an expanding variety of different methods, different technologies, including that reverse genetics that we spoke about earlier, to understand gene function. But not only to understand gene function, to understand the entire network of which includes that gene and other genes that interact for a particular purpose in the organism, and also how the gene products, that is maybe the proteins or the RNA molecules, are also involved in a particular biological process. Because often you find that it's not just one gene and the product of that gene that causes some effect in the organism. It's often an interaction between various genes and gene products, RNA molecules and proteins, really complex networks involved in understanding gene processes. All right, enter the genomics revolution. So following the development of recombinant DNA technology in the 1970s, researchers would typically clone and sequence one gene at a time. And this cloning and sequencing from scratch could actually take several years, just for one gene, really laborious process. Then by the time we hit the 1980s, it was decided that a genome project would be undertaken. So a large team of researchers decided that if they could assemble enough people, enough computing power and so on, that they could actually clone and sequence the entire genome of an organism. And they decided that the sequence and the clones would be publicly available. And of course, if successful, these genome projects would enhance the speed of research efforts. So here began an era known as the genomics revolution. Of course, a genome sequence would provide some sort of insight into how a genome is structured or how it's built. So, for example, it could tell you how many genes are in the 3 billion bases that constitute the human genome. How are those genes distributed and why are they distributed in such a fashion? What fraction of the genome is actually coding sequence and what fraction is non-coding sequence? What fraction of the genome is regulatory sequence? What are the functions of the genes? And how do we actually read the information that's provided in genomes? And I can hear someone trying to get on, but they can't. All right, so the basic techniques for sequencing were already available in the 1980s, but the sheer scale of sequencing an entire genome was actually beyond the capacity at the time. So this was only in the 1980s. And to see what they can do today compared to what they were doing in the 80s is just mind-boggling. It's really difficult to keep up with how quickly the technology is advancing in this area anyway. So these large research centres developed all the robotics and all the automation that were required to carry out the thousands of cloning steps, the millions of sequencing reactions that were required to assemble the sequence of an organism. And the golden age of genome sequencing was then in place in the late 1990s. And the rate of genome sequencing has continued to grow. It's really accelerated. And various methodologies have been invented. They're much faster, they're much cheaper, they have a much greater throughput. So for example, these days, over 500 million bases of DNA sequence per day can be obtained on a single instrument. And this represents more than a 1,000-fold increase over those earlier instruments that were used to obtain that first human genome sequence. But characterising whole genomes is really fundamental to understanding the entire body of genetic information that every living organism contains. All right, so let's define, what do we mean by the sequence of a genome? The highest resolution map of a genome is the complete DNA sequence of nucleotides. That is the complete order of nucleotides, that is A's, T's, C's and G's, of each double helix in the genome. In other words, if you have a complete genome, you would know for every chromosome what the order of nucleotides is in that chromosome. That's essentially what's meant by sequencing a genome. All right, we're going to look at the basic approach to obtaining a genome sequence. Basic approach. Firstly, you need to break the DNA molecules of the genome into thousands to millions of random small segments. And many of these segments will overlap with each other because you're not just breaking the DNA molecules from one cell, but from many, many cells. And they will break in different places. So you'll have overlapping pieces from individual genomes. Then you need to be able to read the sequence of nucleotides of each of those small segments. Then using computer software, find the overlap between those small segments where the sequences are identical. Then continue overlapping pieces of DNA sequence until all those little segments are linked together. At this point, the genome sequence is said to be assembled. Okay, let's look at that process right here. The logic of obtaining a genome sequence. So we start with a genome. So this might be one chromosome, for example, but as I just said, you wouldn't just use one chromosome, you would use lots of copies of that chromosome, that is genomes from multiple cells, to obtain a genome sequence. All right, you would cut the genome copies into random fragments, as you can see here. Then you would sequence each of those small fragments at number 2. Number 3, you overlap those sequence reads. So, for example, probably not a good example here, but let's say this was the original piece of DNA, and in cutting that DNA, this was the original piece of DNA, and in cutting several copies of that piece of DNA into different size fragments, you might end up with one fragment like this, and then another fragment like this, but these two fragments contain at this end, at these ends, exactly the same sequence. And then this is another fragment, which is also exactly the same at these two ends. In other words, you've got these overlapping regions of identity, which means you can actually join them together to get a longer piece of DNA sequence from that original genome. And when you overlap them and assemble them in this way, you have constructed what's called a contig. So this is called a contig. So all those sequence reads are overlapped, and this is why you need such tremendous computing power to be able to deal with, potentially millions and millions of sequence reads, and then have the ability to overlap them to find those similar regions between fragments and overlap them to produce these contigs. Then the contigs themselves, or the individual contigs themselves, are also compared, and where there are ends of those contigs that are exactly the same, they're also aligned with each other. Here we go. Also overlapped and aligned to produce a complete sequence. Now that's the general gist of obtaining a genome sequence. Right, so why does this process require so much automation? Let's start by considering the human genome with 3 billion base pairs. 3 billion base pairs. If we could purify the DNA just directly from each of the 24 chromosomes, so remember we only need to purify and sequence one copy of each chromosome. That's why we're saying 24 chromosomes, and read their individual sequences directly. It would be very straightforward. But we can't do that. That's just not possible. The technology to take a single chromosome and sequence that entire chromosome just doesn't exist. That can't be done. So instead, automated sequencing is currently the state of art in DNA sequencing technology. And initially it was based on the pioneering work of a chap called Sanger, and he basically invented the first sequencing method called dideoxy chain method. But now they employ new chemistries and new optical detection methods in sequencing. And just by the way, these individual sequencing reactions that read, these small fragments of DNA are called reads. And they normally only read each reaction only reads, as I said, just small regions of DNA, about 300 to 600 bases in length. So a major challenge of a genome project is actually sequence assembly. That is taking all those little sequence reads and building them up and joining them together into a consensus sequence. And what do we mean by a consensus sequence? This is a sequence of DNA nucleotides for which there is agreement that it actually is an authentic representation of the DNA. So in other words, you can't just sequence a small piece of DNA and say, okay, that's it, that's the sequence. It has to be done multiple times, just like you would include multiple replicates in an experiment that you were performing to make sure that you've got a genuine result, or as genuine as possible anyway. Okay, so that's a major challenge. But as with any experimental observation, automated sequencing machines don't always give perfectly accurate reads. There can always be problems. You could get results that are not correct. Really depends on the efficiency of the dyes that they use. It depends very much on the purity of the DNA that's been extracted. and also on how intact that DNA is, and it also depends on the specific sequence of DNA. There's certain nucleotide sequences that seem to be really easy to sequence, and there's others that seem to be really difficult to sequence. And that always comes about in sequencing results, where you get good resolution of certain parts of a DNA, a fragment of DNA, and poor resolution of others. All right, so how to ensure that we have a consensus sequence? So to ensure this accuracy, we need to obtain multiple, that is about 10 successful reads of each base pair. So for example, for the 300 billion bases in the human genome, we would need 100 million successful reads to give this 10-fold coverage, which is considered to be fairly accurate. But remember, not all the reads are successful, so the number of attempted reads is actually even larger than that. As we said, the aim of a genome sequence, of a genome project, is to produce a consensus sequence that is a true and accurate representation of the genome of a reference individual. Keep that one in mind, that a genome sequence represents only one individual. So that genome serves as a reference sequence for the entire species. Okay. And we say it's a reference sequence because there are many differences in DNA sequence between different individuals, even within the same species. So no one genome sequence truly represents exactly the genome of the entire species. Okay, because of course they're extracting the DNA for the genome sequence from one or two individuals. All right, so we're going to look at two different approaches. There's actually a few different approaches to whole genome sequencing. We're going to look at two different approaches, just very briefly. So the current strategy in genome sequencing is called whole genome shotgun sequencing, usually abbreviated to WGS, whole genome shotgun sequencing. And the traditional WGS that was used to sequence the human genome relied on the cloning of DNA in microbial cells and employed the original Sanger dideoxy sequencing technique, which we won't describe here. All right, but here are two different approaches that differ in how the short segments of DNA are originally obtained. The first approach is the traditional whole genome shotgun sequencing, and the more recent approach, even though it's actually been around for quite a while now, is called next generation WGS. And that's normally abbreviated to next-gen sequencing. And this particular method employs the use of cell-free methods, so you don't need to use microorganisms, and it's designed for much higher throughput. All right, so let's start with traditional whole genome shotgun sequencing. So it begins with the construction of what's called a genomic library. So this is where the DNA is cut, it's cleaved at specific sequences using enzymes called restriction enzymes, which you will have heard of before, maybe from living systems. So these restriction enzymes, some of them cut the DNA at many places, other enzymes cut the DNA at fewer places, at different sequences, but overall restriction enzyme cutting generates small fragments, small DNA fragments with overhangs, with those sticky ends that we've also learned about in living systems. And then those short DNA fragments are inserted into some sort of accessory vector. Some sort of vector, and in particular a vector that can be inserted into bacteria or yeast and then replicated to produce lots and lots of copies of that little DNA fragment. All right, so some common vectors that are used. Plasmids, you've heard about. They can use modified viruses and artificial chromosomes, bacterial artificial chromosomes, yeast artificial chromosomes, and others. Oh, and by the way, they call it shotgun, whole genome shotgun sequencing, because the clones are actually randomly obtained without any idea of where they fit in the genome. So very random process. All right, so the short DNA segments are inserted into these plasmids, for example. That is, they are ligated into the plasmid, which has also been cut with a particular restriction enzyme. So here's a description here of how they make the genomic library, which I've just been talking about. So let's go through this. Restriction enzymes are used to cleave the DNA into short DNA segments. Then those short DNA segments or fragments are ligated to the DNA of the vector. So for example, the plasmid. Then these are propagated, that is, lots of copies are made by introducing those vectors into bacteria, and then the resulting clones are sequenced and then overlapped to generate contigs. And we can see that here. So here we have, for example, one plasmid vector. Here is your fragment, here is your fragment in green that you cut from the DNA, and you've inserted that into the actual plasmid here. That plasmid's been inserted into bacteria to generate lots of copies, because you need a certain amount of DNA before you can sequence it. Sequences are very powerful, but they're not that powerful that they can sequence the DNA from one single plasmid. You would need lots and lots of copies of this. Right, so this is your little cut piece of DNA that you've ligated into the plasmid, and then in order for the sequence of that DNA fragment to be read, you would need to use primers, which you can see here. And in order for a primer to work, you have to know the sequence that that primer will bind to. Just as you did with your PCR reactions, you used primers with known sequences, so those primers would anneal to your plant DNA or your fish DNA or grapevine DNA. All right, so because we don't know the sequence of that little segment of DNA that's been inserted there, that's what we're trying to find out. So the primers are designed to bind to the complementary DNA in the plasmid vector itself, because we know exactly what the sequence of the vector is. So you can see this primer here will bind to complementary DNA sequence here. And then when the sequencing reaction begins, it'll start forming complementary DNA to your unknown DNA fragment here. And the same thing will happen with this primer here. So you'll end up with generating a piece of DNA here that will be read, the sequence will be read, and a little piece here of your unknown DNA fragment that will also be read and sequenced. All right, so the output from this is a very, very large collection of these random short sequences. And some of them will be overlapping. And those that are overlapping can be assembled into a consensus sequence that will cover the whole genome, or I should say most of the genome. That is by matching those overlapping sequences. And those overlaps are assembled into those sequence contigs that we mentioned earlier, where contig is referring to a contiguous or touching sequence. That's what we mean by contig. All right, so the primer that we have at this end of your unknown green DNA segment can generate a sequence of about 600 base pairs, and the same for this one. So when you get a sequence at both ends of that unknown DNA fragment, that's referred to as paired end reads. So you don't get the bit in the middle, depending on how big this little fragment is, but you might get a bit at either end. Paired end reads. All right, so let's look at this new approach, or relatively new approach at sequencing. And this approach was developed by the 454 Life Sciences Corporation. It is called Next-Gen WGS sequencing, next generation sequencing. It employs new methodologies that really increase output. Let's look at this, and then we'll look at an image that shows it very nicely. So in step number one, the DNA molecules are prepared for sequencing in reactions that don't require any cells. So you don't have to use any sort of microorganisms to clone your DNA molecules for you. Number two, millions of individual DNA fragments are isolated, and then they're sequenced at the same time during each machine run. Number three, advanced technologies make it possible to actually detect that sequence in really, really small volumes. All right, so this particular type of next-gen sequencing is called 454 sequencing because it was developed by the 454 Life Sciences Corporation, and it's also referred to as pyrosequencing. So in stage one, a DNA template library of single-stranded DNA is constructed. Single-stranded DNA. In stage two, those DNA molecules are amplified by polymerase chain reaction, PCR. But before they're amplified by PCR, they are immobilized on very tiny beads. Then in stage three, a novel sequencing chemistry called pyrosequencing is employed. So in this pyrosequencing, our enzyme called DNA polymerase, which you might remember is the enzyme that is used in DNA replication. So this enzyme and primers are added. Each of the 4 nucleotides are added, and these will flow through each of those little wells one at a time. So the 4 deoxyribonucleotides, of course, you're familiar with. Then when each of the nucleotides are incorporated into that complementary sequence of DNA that's being synthesized, then a pyrophosphate is released. And you might remember your pyrophosphate, PPI, we abbreviate it to PPI. And that's also something that goes back to one of your living systems pracs on enzymology. Anyway, when the pyrophosphate is released, then 2 enzymes called sulfurylase and luciferase will convert that pyrophosphate signal to a visible light signal. That light signal is recorded, and that will tell you whether the next nucleotide in that sequence of DNA is an A, C, T, or G. So let's look at that illustrated, on the next slide here. All right, so just looking at the left, the image on the left, and this is showing these tiny beads on which those pyrosequencing reactions will take place. So single strands, single DNA strands are attached to individual beads. Then in number 2, those single strands of DNA are amplified using PCR. And the single strands, the amplified pieces of DNA, the new pieces, remain attached to those beads. Then each bead in 3 is deposited into a tiny well, 1 bead per well. And this is what the wells look like, even though you can't actually see them here because they're so incredibly small. And then here we can see the chemistry of the pyrosequencing reaction. So here's the bead. Here's one of the attached single-stranded DNA segments. And then when the pyrosequencing reaction begins, the DNA polymerase enzyme will incorporate nucleotides that are complementary to that original strand. So you can see the reaction is proceeding in this direction, where the red arrow is pointing. Here we have our enzyme at work, our DNA polymerase enzyme. And up here we have repeat additions of the 4 nucleotides, that is A's, G's, C's, and T's. And they are passed through each of these wells to facilitate this reaction here. So if we look at this one here, here's a D-GTP, so this is a guanine, but it's a guanine triphosphate. And that's incorporated here in the next step because it's complementary to the T in the original DNA segment. And when that's incorporated, that's when the pyrophosphate PPI is released from it. Then we have these enzymes coming into play. Sulfurylase will incorporate that pyrophosphate into ATP. You'll remember your adenosine triphosphate. And then this enzyme, luciferase enzyme, will actually convert that to a light signal. And the light flash will indicate that the guanine was added to the new strand. And that's the basis of our 454 pyrosequencing reaction. Very elegant technology right there. All right, so we said that whole genome sequence assemblies are quite difficult, and that is certainly the case. And the difficulty of assembling a genome really depends on the complexity of the genome. So for example, bacterial genomes tend to be really easy to assemble because they're generally single copy DNA and they don't have all those repetitive DNA sequences that you find in, for example, the human genome. And for that reason, as of May 2010, over 600 bacterial genomes had been sequenced. Now, with eukaryotes, as we just said, there are numerous repeated sequences in eukaryotic genomes. And as you know, that's still unknown as to why there's so much repetitive DNA in the genome. So that's a big stumbling block in whole genome assembly of eukaryotic sequences. All right, but once the genome sequence of a particular organism is complete, then it actually becomes much, much easier and cheaper to sequence further individuals within that same species. So we'll look at an example here. So in 2008, sequencing of the genome of a famous scientist, James Watson, was completed. And you might remember Watson and Crick. They were the 2 scientists that actually discovered the structure of the DNA double helix. Okay. So I probably should mention here that the 454 pyrosequencing method that we just covered, it's also referred to often as massively parallel sequencing. And the reason for this is that they can run several 100,000 to over 1 million reactions at the same time. And that's why it's said to be high throughput, because the older machines could only run 384 reactions at the one time. So that's a huge difference, 384 versus more than 1 million reactions simultaneously. So this is called massively parallel sequencing. All right, so genome sequencing of the famous James Watson was completed. There were 100 million sequence reads that were performed, representing a 7.4-fold coverage of his genome. The cost was just under $1 million, which sounds incredibly expensive, but this represented less than 1/100th the cost of the first human genome. So in just 7 years or so, the cost had come down right down to less than $1 million. Less than $1 million. Okay, what about these days? So the prediction was that eventually it would only cost $10,000 per human genome or even less. The current cost of sequencing a genome of an organism is about $5,000 to $6,000. That's how cheap it's become in only the last 10 years or so. And that $5,000 to $6,000, that won't give you a complete coverage, but a very good coverage of the genome. All right, so let's look at bioinformatics. The study of the information content of the genome is called bioinformatics. Information in the genome can be thought of as the sum of all the sequences, all the DNA sequences that encode proteins or encode RNA molecules, plus all the binding sites that govern the time and place of their actions. So the principal objective is the identification of all the functional elements of the genome, and this is referred to as annotation of the genome. And keep in mind that we are very, very far away from being able to read and understand this information in any one genome. There are some genomes that are quite well annotated, well, relatively well unannotated, and others that are really poorly annotated. So, for example, the model plant Arabidopsis, its genome has been studied extensively since it was first sequenced and is relatively well annotated. And then we've got something like the wheat genome, which was only really released in its completeness, well, almost complete, not long ago. And if you try to work with that genome, then you'll be confronted with very, very poor annotation. So really difficult to work with. All right, here we can see an illustration of the information content of a little part of the genome. So what we can see, so what can we see here? What sort of information do we need to know about a genome? And this is just one gene represented here. Here it is, one single gene, and you can see that really to understand the genome, you have to know where the introns are, where the exons are, what are the codons within the exons. When we're talking about the introns, where are the binding sites for this complex called the spliceosome that will actually cut out those introns from the RNA during RNA processing? Where is the translation termination binding site of that gene? What part of the DNA represents the 3' untranslated region? What part of the genome represents the formation, or where the poly A tail will form. What part of the genome represents where the ribosome will bind to the messenger RNA? What about the sequence of DNA in the 5' UTR, untranslated region? What about the promoter region? That's always an unknown. How big is the promoter of a gene? Is it only 500 base pairs long? Is it 2,000 base pairs in length? And of course, within the promoter region, what is the sequence where the RNA polymerase enzyme binds so that it can start transcribing the actual gene itself? And then what about all these regulatory elements? There are lots of regulatory elements, and these could be very close to the promoter region. They could be really far away from the promoter region. So you can see already in just within this one gene and around this one gene, there's lots of information content right there. And this is not even all of it. So when we consider a single gene, as we've just considered here, we can view it as a series of binding sites for different proteins and even different RNA molecules. So lots of information that needs to be deciphered from a genome sequence. All right, and once you do identify or predict, I should say predict, where the genes are within a genome, which ones are the actual protein coding genes? Because we know that not all genes encode proteins. There are genes that encode RNA, various RNA species, and you'll be familiar with ribosomal RNA and transfer RNA, but there's also lots of different ones, for example, small nuclear RNAs and so on. And because the proteins, it's the proteins in the cell that largely determine the morphology of the cell and all the physiological and chemical properties of the cell, one of the primary aims in analysing a genome and annotating a genome is firstly to try to determine an inventory of all the polypeptides that are encoded by an organism's genome. And this inventory is referred to as the organism's proteome. So in order to determine this list, the sequence of each messenger RNA molecule has to be deduced, because as you can imagine, if that gene is a protein coding gene, it is going to be transcribed into messenger RNA, because you know that it's the messenger RNA that determines the polypeptide sequence. So if you can sequence the messenger RNA molecules from a cell or from cells, then you can be fairly sure that those messenger RNAs will be translated into proteins. And that enables you to determine the proteome of the organism. All right, challenges to working out a proteome is that many of the eukaryotic genes actually have introns. It's now known that many genes have alternative exons. So what you might identify as being the exon or the exon regions without a gene, these can change depending on environmental conditions. So a gene can be expressed in one way and having a set number of exons, but when that same gene responds to certain triggers, certain stimuli, it might actually change the borders of those exons and be transcribed in a very different way. And this is referred to as alternative splicing. Identifying the 5' and 3' splice sites directly from the DNA is actually quite difficult. All right, so here are some approaches that have actually been used to determine which genes are protein coding genes. All right, so let's start by firstly defining this term here, ORF. ORF So an ORF is, this is what's referred to as an open reading frame. And an open reading frame is composed of start and stop codon of a gene, and then everything else in between. That's referred to as an open reading frame. And these open reading frames can be determined to a certain point by certain computer software. And that software has been designed to look for DNA sequences with certain characteristics that are typical of genes. As I said, they look for particular start codons and stop codons to be present. They look for the gene size. So if you have a start codon and then after 10 base pairs, you have a stop codon, the software would discard that. It wouldn't actually recognise that as a gene sequence. So it has to be of a particular size. And it has to be composed of sense codons as well. All right, so that's one approach. Here's a second approach. And that is to detect these open reading frames that will potentially constitute genes, protein coding genes, and this involves analysing the messenger RNA expression. And the way they do that is to take the messenger RNA, copy it into DNA, which is called cDNA, and then basically create libraries of those cDNA sequences. And if you can actually synthesize cDNA from messenger RNA, then this provides actually direct evidence that a given sequence is expressed and therefore represents a true gene. So this is generally termed to be the gold standard proof. All right, there's also large sets of cDNA molecules available for which only the 5' or 3' ends or both ends have actually been sequenced. And these are referred to as ESTs, or expressed sequence tags. So these expressed sequence tags can actually be aligned with the genomic DNA itself to actually determine the boundaries of the actual gene, or the boundaries of the transcript. All right, and so here this is illustrated for you. So here we have the genomic DNA showing the exons and introns in between. This is what we would call the cDNA, complementary DNA or copied DNA. This is what it would look like. So the cDNA, you can see, is much shorter than the genomic DNA, of course, but also what's missing are those introns. The introns are missing because this cDNA has been copied from the messenger RNA, that processed messenger RNA, which has already had the introns spliced out. So that's the cDNA. So imagine all these exons are actually joined together in the cDNA molecule. And then we have the final one, ESTs, expressed sequence tags. So these represent just the end sequence, the 3' sequence of a gene, and maybe the 5' sequence of the gene. Okay, so they're called ESTs. All right, so then using computer programs and databases, which anyone can access, you can actually compare sequences, your sequences of your DNA fragments with all other sequences that have ever been found. And these public databases will actually give you predictions of binding sites that will search for very highly conserved DNA promoter sequences and transcription start sites, splice sites, and so on. All right, and here's a couple of examples of the databases, or rather the programs within those databases that are used, BLASTN or BLASTP. A really quick, easy way to try and get some sort of idea as to what that gene sequence, for example, what family it might belong to, or whether it has any really highly conserved sequences in there. And it can even, these websites, these databases can even give you an indication of the three-dimensional structure of the protein that would be encoded by your DNA sequence. Very useful. Did you do any work with NCBI, with an NCBI database? With PIPA? I'm not sure if you do. No? Doesn't ring a bell? No? Okay. All right, and here we have genome searches hunt for various binding sites in the DNA. All right, so this is very similar to a previous image that I showed you. So, and this represents different sources of information that can be combined to create the best possible messenger RNA and gene prediction. So using information from cDNAs, from ESTs, from those public databases, and so on. And that all can be used, all that information can be used to create this map of different binding sites within the DNA. All right, let's just turn our attention to one of our model organisms, the plant model organism called Arabidopsis thaliana. And this plant has been used for over a century as a model organism for scientific study. And of course, you didn't get to extract DNA from Arabidopsis from memory. But it's normally very, very easy, very straightforward to extract DNA from. And it just so happens that it's now growing very happily in the lab in G118. All right, so let's look a little bit at the snapshot of the structure of the Arabidopsis genome. So as of 2014, and these numbers would have changed since then, the Arabidopsis genome contains, well, this won't have changed, 10 chromosomes. That is the diploid number of chromosomes in Arabidopsis. 27,000 protein coding genes, 4,800 pseudogenes, or transposable elements. We'll talk about transposable elements in the next lecture. Pseudogenes are simply false genes. They have all the characteristics of genes, but they don't seem to do anything. So they're called pseudogenes. Over 1,300 non-coding RNA genes. 33,000 genes in all. So there we go. And it was also found that about 18% of Arabidopsis genes are known to have different, what they call splice variants. That is, those genes can be expressed in different ways. That is, different lengths of transcripts and so on. All right, let's move on to comparative genomics. This is one of the most powerful ways to analyse a genome, and that is to compare DNA sequences between related species. All right, functional DNA sequences tend to be conserved, very conserved, over long periods of time, as we saw in our lecture last week. And genes identified in one species are likely to be identifiable in related species, sometimes even in very unrelated species. And comparative genomics can also reveal how species change, how they diverge as different species evolve and change through changes in DNA sequence. Okay, so it's very interesting that when one particular gene is first studied, and that maybe the function or part of the function of that gene, the regulation of that gene is actually identified, and then they go on to use comparative genomics to see if that same gene or a similar gene is found in other organisms. I think in every case where that's been done, they will find similar genes in different organisms or in other species. In particular, in plants, there was a gene that they studied years ago, that they identified years ago. They did some study on it, found that it was involved in pollen fertility. It was critical in fact for fertility of pollen, the viability of pollen. And then they decided to use this comparative genomics, and they found almost exactly the same gene, not just in Arabidopsis, but also in canola, rice, wheat, spinach. And not only that, the gene performed the same function in all those different crop species, as well as that model plant Arabidopsis. So really powerful, the use of comparative genomics. All right, let's look at a really interesting example of where they actually used comparative genomics to compare the DNA from non-pathogenic E. coli and a pathogenic strain of E. coli. So we're all familiar with E. coli, Escherichia coli. It's normally benign. It's a symbiont, that is, it's found in huge numbers in the gastrointestinal tract, not only in humans, but in lots of different animals. All right, so it doesn't normally cause any problem. In fact, it's very useful as one of our gut bacteria. All right, enter a model organism. That is a lab strain of E. coli. And in fact, this is the strain of E. coli that we use in the lab. It's called K12. And the genome of E. coli K12 was sequenced, and it was found to have 4,405 genes. All right, let's go back to 1982. There was an outbreak of infection due to undercooked ground beef that was found to be contaminated with E. coli, but a particular strain of E. coli called O517H7. And this particular strain of E. coli is a pathogenic strain. It can cause some pretty serious infections, and in particular, a fraction of people that are infected with O157 develop a disease called hemolytic uremia syndrome. And in this disease, the red blood cells start being destroyed and the kidneys can fail. So this is potentially a life-threatening disease. And this particular disease is actually the principal cause of kidney failure in the US, with an estimated 75,000 cases of E. coli reported annually. Okay, so let's think about what the difference is between just a normal strain of E. coli, harmless strain of E. coli, and this pathogenic E. coli 0157H7. And of course, you might have heard occasionally, even around Australia, that local swimming pools get closed down. And sometimes that's because there has been some illness, some reported illnesses, severe illnesses from pathogenic bacterial infections. All right, so to try to understand the genetic basis of this pathogenicity of O157, the genome was sequenced. and they found that there were 5,400 genes in the pathogenic E. coli compared to 4,400 genes in the non-pathogenic K12 strain. They also found that, of course, there were 1,387 genes in the pathogenic E. coli that were not found in K12, and there was 500 or so genes in K12 that were not found in 0157. So they actually mined these genes, studied these 1,387 genes unique to pathogenic E. coli, and what did they look for? They looked for virulence factor genes, and virulence factor genes will produce toxins, they'll produce cell invasion proteins, they'll produce proteins that will break down cells, kill cells, adherence proteins, proteins that enable the E. coli to actually attach to cells so that they can invade. Genes that encode antibiotic resistance. So, of course, these pathogenic bacteria don't respond to antibiotic treatment, and so on. Okay, and here we have E. coli 0157 compared with our normal K12. All right, so this represents the one chromosome of E. coli, and these little blocks here represent islands of genes that are specific to each strain, each of those two strains. All right, and finally we move on to functional genomics. So functional genomics involves a global approach to study function, expression, and interactions of gene products. All right, in addition to the genome, there's also other global data sets referred to as omes, such as genome, of course. So following the example of the term genome, so meaning all the genes, researchers have also coined these other global data sets. So here's the term transcriptome, which means sequence and expression of all the transcripts in the genome, where they are, when and how much. We've already looked at the proteome, and there's also others. There's the interactome, there is the lipidome, looking at lipids, epigenome, metabolome, variome. There's, quite a few. In fact, it's very difficult to keep up with all the omes, all the data sets, the omes. All right, so how do they actually study a transcriptome? That is a collection of all the RNA transcripts that you would find in a cell. They use something called, or they used to use something called DNA microarrays, and these are just recently starting to be replaced by, once again, new technology. So these DNA microarrays can answer the question, what genes are active in a particular cell under certain conditions? That is, at what stages in development of the organism, what genes are active in the presence of a particular hormone or in the absence of a hormone, what genes are active or not active in the presence of a pathogen, what genes are active in this plant when it's exposed to a particular abiotic stress, such as heat stress, drought stress, frost, and so on. So really useful technology. So in this DNA microarray technology, and the microarray is often referred to as a chip, samples of DNA are laid out as series of spots which are bound to a glass chip which represents most or all of the genes in a particular genome. And I don't have an image of a microarray chip for you, but that's okay. So just very briefly, and you don't have to remember this is just for your interest, the microarray is exposed to cDNA probes, and it will include a control and a test. Then we have fluorescent labels attached to the probes. In step 3, the relative binding of the probes is detected by a laser beam illuminated microscope. And then gene levels of expression, genes whose levels of expression are increased or decreased under a certain environmental condition are identified. So essentially the output that you get can look like this down here. So where you see those red spots, that means that those genes, the expression of those genes have increased in response to a particular stress, for example. And the green spots represents genes whose expression decreased in response to a particular stress. And then the black ones, they seem to be expressed at the same level regardless of whether you applied a particular stress or not. They're quite a useful experiment and very expensive. All right, just for your interest, here are some more techniques that have been commonly used in genomics. So here's a technique called a 2-hybrid test, which is used to study which proteins bind to other proteins. In other words, the protein-protein interactome. Then there's another technique called the 1-hybrid technique, which will study which proteins bind to DNA. So the study of protein-DNA interactions. And here's another one, the chromatin immunoprecipitation assay, abbreviated to a CHIP assay. And this enables the study of the protein-DNA interactome. As I said, you don't need to remember those. All right, and finally, I believe we're nearly finished with this lecture. A little bit more on reverse genetics. So how do you determine gene function? So in this process of reverse genetics, you would disrupt the DNA sequence somehow. You would mutate or interfere with the DNA sequence, or the messenger RNA, or the protein. So in doing that, you're going to cause some sort of mutant phenotype, some sort of problem with the plant, for example. And when that happens, then you can attempt to assess the role of the normal gene product in the biology of the organism. So if you disrupt a particular gene sequence and you find that that impacts on the shape of the leaves, for example, then obviously that gene has something to do with leaf patterning. If you mutate a particular gene sequence and that, that plant, for example, can't photosynthesize properly, then obviously it plays a role in the photosynthesis machinery and so on. So there's several approaches that are used to interfere with gene function for the purpose of reverse genetics. And here are 3 listed: random mutagenesis, targeted mutagenesis, and RNA interference, abbreviated to RNAi. All right, and this just illustrates that process of RNA interference for your interest. All right, I think it's time for a break.
Yep.
Who agrees?
Yeah.
That's probably a lot to take in, isn't it?
Yeah, it's...
Here we go. All right, welcome back. The very last lecture for Applied Genomics and Plant Breeding. And we're going to talk about the fascinating world of transposable elements. And here we go. All right, so in the 1930s, there were some studies performed, some genetic studies performed in maize that really upset the classical view that genes are fixed within chromosomes. And this research that was conducted suggested that some genetic elements can move from one location to another within the genome. That is, they can move to different positions in the same chromosome or into different chromosomes. And these elements were actually detected by the mutations that they cause when they insert into other parts of the genome, and that results in an activation or inactivation of a gene. And these were found in Drosophila, in the early days in Drosophila, maize, yeast, the nematode worm, Caenorhabditis elegans, and even in E. coli. But we probably should consider or remember that these findings at first were viewed with a lot of doubt for many years. But research has since shown conclusively that there are some genetic elements that can move from one location to another. So these mobile elements have been given a variety of names, such as jumping genes, controlling elements, mobile elements, transposons, and transposable elements, often abbreviated to TEs. They're now known to be very widespread in nature, so they're found in microorganisms, plants, and animals. They actually constitute the largest component of the human genome. About 50% of the human genome is thought to be made up of transposable elements. So even though these TEs have been found in virtually all organisms studied, their genetic role is still unknown. All right, so let's go back to maize or corn, or Zea mays. It's actually a member of the grass family, and along with rice, wheat, and barley, these are the most important source of calories for the human race. Let's go back a little bit further, back to the 1920s, and this is where a scientist called Rollins Emerson set up a lab at Cornell University to study the genetics of corn traits, including kernel colour. And corn, keep in mind, corn was very widely studied in genetics, firstly because the male and female flowers are really well separated, physically separated, which made genetic crosses really easy to carry out. And also, the large chromosomes that you find in corn makes it an ideal organism in which to study cytogenetics. That is to study the analysis of chromosomes. That's called cytogenetics. Now, in the 1940s, a member of Emerson's lab, a young female geneticist called Barbara McClintock, she made quite an astonishing discovery. What she noticed was that chromosome number 9 frequently broke at one site, and you know that a site on the chromosome is called a locus, or a locus. Some people say locus. She determined that that breakage was due to two genetic factors. One was called a DS for dissociation, and this DS element was located at the site of the break. And the second element, AC for activator, was actually required to activate the breakage at the DS locus. All right, so Barbara McClintock suspected that A, C, and DS were actually mobile genetic elements, and she suspected this when she couldn't actually map AC. That is, in some plants, she mapped, that is, she identified the location of the AC to one position, to one locus, while in other plants of the same line, the AC element mapped to different positions in the chromosome or in another chromosome. Also, she observed there were rare kernels, which you can see pictured here, with different phenotypes that appeared. So one phenotype was a colourless kernel with pigmented spots, as shown here. All right, so here we have a description of the phenotypes of the chromosome breaking strain of corn compared to the phenotype of one of the derivative strains from that chromosome breaking strain. But rather than just reading through this, let's actually look at it right here. And just note that this is a really important slide here. And of course, the slide before and after, which describes what's going on. This is really important for your final exam. So take note of that. All right, so let's start with the chromosomes here, this pair of chromosomes that we can see in A. And let's describe this. This is a pair of 2 copies of chromosome number 9. And let's look at the alleles that we have here. So we have this C allele, and the C allele, the normal wild type allele, is the allele for pigmentation. Okay, so when that allele is active, which it is in this case because the dominant allele, it's abbreviated to C, that results in pigmented kernel. Our next allele, SH, capital with a capital S. So this one is the allele that causes the kernel to be plump. Okay, next one along, WX. This is the normal wild type allele, which gives the kernel a shiny appearance. Okay, so that's all good. They're all dominant alleles. And just note what's inserted here, the DS, the dissociation element. Okay, and it's just sitting right in there. All right, if we look at the other copy of chromosome 9, that is the homologous chromosome, it has recessive alleles, the recessive alleles. And you can see they're recessive because they're all shown here in lowercase. So let's look at what happens if these were actually expressed, these recessive alleles. The little c results in a colourless kernel. And that's what we're seeing here, represented by this coloured portion of the kernel. Little sh represents shrunken kernel, in contrast to the normal plump. And then the little wx here represents part of the kernel that is not shiny. Okay, so that's if these alleles were being expressed, which in this case they're not, because just one normal or wild type copy of each allele is enough to produce a pigmented kernel, which is plump and has a shiny surface. All right, now let's look at what happens when this DS element is activated by the AC element to break the chromosome. And that's what we can see in this part here. Here it is here. All right, so the AC activates chromosome breakage at the DS site. In other words, these wild type alleles, dominant alleles, have been lost because this whole fragment from the chromosome has been lost. All right, now we have an expression of the recessive alleles. An expression of these recessive alleles results in colourless, shrunken, and not shiny sector in the corn kernel. In other words, when this kernel was first developing, one of the cells in which this chromosome breakage occurred, that cell would divide to produce more cells in the developing kernel, and each of those daughter cells would also have only part of this chromosome 9. So all the cells in the kernel derived from that original cell that had the breakage in chromosome 9, all those cells derived from that original cell would have this phenotype: colourless, shrunken, and not shiny. So hopefully that makes sense to you, and we'll go over it again in just a minute. All right, and now we've got a derivative of that original line, and this derivative produced new unstable alleles. Well, one in particular. All right, so we've got the same sequence of alleles here, dominant alleles, same sequence of recessive alleles on the homologous chromosome number 9. But in this case, the big C actually has the DS element inserted into it, inserted into the big C gene. And that means that the big C is not expressed, and that results in a completely colourless kernel, completely colourless background. But then if AC actually activates the DS loss from that big C gene, and this is what's happened here, the DS element moves out of the big C gene, then that will restore pigmentation in all the cells that are derived from the original cell in which the DS was lost from the C gene. And that results in all these little patches of pigmentation in an otherwise colourless kernel.
Silvana?
Yes.
So as it grows, is it just going to become more pigmented, or will it eventually become so much pigmented like into the first image where it's almost, you know?
No. This is a different line that's actually produced this.
Okay.
A different set of genetic conditions. So if you can think of this as, and I probably should draw it, if you can think of this just as, for example, just a few cells.
Yeah.
So very, very early on in development of this kernel, just a few cells. Let's say, I think if I just draw 9 cells, that will be enough. Just a few cells, and all of them have this this genetic background here, this genotype here, I should say. All of them have this genotype at the 9 cell stage, right? But let's say that during that stage, this happens. That is, the DS, the AC activates DS loss from the C gene, and let's say that happens here in this cell, and it happens here in this cell here. Okay, then when all these cells keep dividing by mitosis to form this complete kernel, then all the daughter cells that are derived from this one
will have the pigmentation.
Will have the pigment, yes. They will all be pigmented, and all the daughter cells derived from that cell there, they will also have the pigment. Yeah, they'll all be pigmented.
The cells where the original activator bound with the dissociative site will have the pigment.
Yes, that's right.
Yeah, all right.
Sorry, all the cells that have the DS lost from the pigmented gene. Is that what you were saying? Yes, that's right. Yeah, where the DS has been activated to leave the pigmented gene, C, only those cells will be pigmented. Okay. So obviously this event has occurred here early on in the development of the kernel, and it's occurred there and there, and there. And that's what's giving rise. And we could think of it just very simply as being one cell. The event occurred in one cell that gave rise to that pigment, and that spot there and that spot there. But it could have been like several cells within this same area.
Okay.
In which that happened. We don't know. We would have to actually look at that much earlier on in development. Yeah. Does that make sense?
Yeah, all good now.
Yeah. Oh, okay. Very
Yeah,
good.
I wanted to know how that worked from the very beginning.
Yes, it's easier to visualise if you can think of it back to, you know, those early cells.
Yeah.
Yeah, and the same thing applies to this one here. Same thing, exactly the same thing applies here. But obviously there's just one colourless region here, yeah, which results from all the progeny of the cell in which this occurred. Okay, whereas the rest of the kernel, so all that coloured part of the kernel with the other phenotypes that go with it, all have this genotype up here. Okay, does that make sense?
Yeah, all good.
Yeah, all good.
Yeah.
All right.
Is
So
that just a random process, Savannah? Or
yeah, it's thought to be well, as far as anyone knows, it's just a random process.
Yeah. Okay.
A random process. I would say that there are triggers to this process, but as to what those triggers are, who knows?
Yeah. Okay.
Okay, so let's go back and now let's just go through this now that you've seen how it actually looks. All right, so in that first example, that top example we looked at, that is in that chromosome breaking strain, a chromosome that breaks at or near the DS loses that end of the chromosome that contains those wild type alleles of C, SH, and the WX genes. When that happens, the recessive alleles on the homologous chromosome are expressed, and that produces the colourless sector in the kernel. Okay, so here's the colourless sector here. But not only colourless, expression of those recessive genes also results in these phenotypes here, shrunken and not shiny. But they were really focusing on colour, kernel colour. All right, and then at the bottom of that slide, we looked at a particular derivative, which affected the expression of only a single gene, the C gene, the pigmented gene. And we saw that the DS was actually inserted into the C gene. And when the AC activated the loss of the DS from the C gene, then all the mitotic descendants from that gene would have colour expressed again, and that produced that spotted phenotype, in an otherwise colourless background. So in other words, this genotype here, where the DS is inserted into the pigmented gene, into the C gene, gives rise to this colourless kernel, the colourless background. But then in cells and the descendants of those cells in which the DS is lost from the C gene, in other words, the function of the C gene, that is the pigmentation is restored, and that gives rise to these spots, these coloured spots in the kernel. All right, so Barbara McClintock hypothesised that DS had moved from a site into the C gene, and that prevented the expression of C, and that caused that colourless background of the kernel. And what caused the spotted parts of the kernel? Let's go over that again. So this unstable phenotype resulted from the movement or transposition of that DS away from the C gene. This allows the mutant phenotype to revert back to wild type and produce those pigment spots in that cell in which it happened and in all the descendants from that cell. So you would have larger spots of colour obvious. If the DS leaves the C gene early in the kernel development, and the earlier it leaves, then the more mitotic descendants there will be, and that gives rise to bigger spots within the actual kernel. So quite an amazing discovery, and Barbara McClintock is probably one of the most famous female scientists of all time. All right, let's look at this, keep revising and introduce a little bit more information. So the DS element inactivated that C gene, the pigmented gene, by inserting into its coding region. The allele that carried the insert is called the CMDS allele, which stands for C mutable DS. A strain that has the C mutable DS but no activator element, no AC element, will have just completely colourless kernels because the DS cannot leave on its own without the AC present. A strain that had the C mutable DS and the activator as well had the spotted kernels, just like the ones that we saw, because the AC activates the DS in some cells to leave the C gene, thereby restoring that pigmented function. The leaving element is said to be excised, or it transposes, and hence that term, transposable elements.
Sorry, Silvana.
Yeah.
So the AC basically initiates the whole process in the first place.
Yes, correct.
In the presence of the DS, but it
Yes.
does. So it will still be there. I guess once it's there to initiate it, will it still be there to then go back to get rid of the DS? Is it?
Yeah, I'm not sure I follow the question.
Yeah. I don't think I explained myself that well.
That's all right.
So it activates the DS initially, so that the homologous C is then active.
Yeah.
But once it does that, will it still be there to activate the DS, or?
Okay. All right. So I think what you're asking, so the AC, you're saying the AC activates
up the top. So up the first, it's there for the first bit to.
Oh, up here. So here's the DS up here.
Yep. So the AC will activate that.
That's right.
Yeah. So
the AC will activate that. Yep. To break the chromosome here.
Yeah. But yeah, that's all. But the second, so B is a new cell.
That's right.
So it will have nothing to do with.
Oh, B is actually a new, not so much a new cell, but a new strain or a new line that originated from this plant line that had the chromosome breaking phenotype, if you like.
Yep.
Yeah.
Yep. Okay.
Yeah.
So if it was in the first one, is there any guarantee that it will be in the second one, or?
No, there's no guarantee that the AC element is actually there.
Okay, so it doesn't get transported to, or it doesn't get moved on to other segments?
Well, that depends on whether it actually, and I'll show you, I think on the next slide.
Yep.
Yeah. Oh, yes, yeah. This might answer. Yeah, let's go through this one. This might actually answer your question.
Yep.
I think I see what you're referring to. Yeah. All right, let's see if this helps.
Yep.
So here we have just the normal wild type C gene in orange here. And of course that produces this phenotype, a pigmented kernel. Then we have, here's another example. This is where we have the CMDS. Here it is, CMDS. In other words, we have the DS element in green inserted into the C gene, into the pigmented gene, but there's no AC present in this line. There's no AC present. So what happens, because the DS is inserted into that gene, it disrupts the function of the gene, so it can't produce any pigment, and you have a completely colourless kernel. And keep in mind that spots, pigmented spots, never develop because there's no AC present in the genome to activate DS to move out of that gene and thereby restore function, which would produce spots in the kernel. Okay, so that's that line. Then we have a different line here. Same thing, we have the DS inserted into the gene, into the C gene, thereby disrupting it. And that produces a colourless kernel, but we have also present in that genome the AC element. And here it is sitting in, it could be either in a different chromosome or a different part of the same chromosome. And with that AC present, if that activates the DS to leave the C gene, the normal function, wild type function of the C gene is restored in those cells in which that occurs. And those cells and the descendants will produce normal pigment, giving rise to those spots. Okay, and in some cases, this is a new one for you. In some cases, what McClintock found was CM with AC. So here we have the activator element itself inserted into the C gene and thereby disrupting the actual wild type function of the C gene. Once again, that would cause a colourless kernel background, but the AC can activate itself to leave, thereby restoring the normal function of the C gene and giving rise once again to pigmented spots. So did that sort of answer your question?
Yeah, look, yep, pretty good.
Yeah. Okay, good. Okay, so that's our phenotypes. All right, and that's explained for you here. So in the CMAC, which was the last example that we looked at, the AC element itself was inserted into the C gene, and this was always unstable. They also found that rarely an AC element could transform into a DS element, just to confuse us completely. All right, other transposable element systems were also discovered and given different names. There was one called dotted, there was another one called the suppressor mutator or SPM system, and that was also known by different names such as the enhancer inhibitor system because different research groups discovered this transposable element system at the same time. Transposable elements were also eventually discovered in bacteria and other plants and also in animals. All right, just a couple of definitions for you. So that AC element and SIMA elements that were also discovered are independently mobile. So they contain all the information necessary for their own movement and for the movement of other elements in the genome. So these elements are termed autonomous elements. Non-autonomous elements, such as the DS element and others that were discovered that were very similar, obviously are not independently mobile. And we've got a nice example here of transposable elements at work in a rose. So what we're seeing here in terms of all these different colours is termed mosaicism. And these different colours are caused by the excision of transposable elements. So insertion of the transposon disrupts the pigment production, resulting in a white flower background, and then excision of the transposable element restores the pigment production, and that results in these red, pink tissue sectors in the rose. So at first the question was raised, do transposable elements only exist in maize? And we need to keep in mind that the other geneticists at the time were very reluctant to consider that similar elements existed in other organisms, because that would imply that genomes are unstable and dynamic, and that was a view that was very inconsistent with the belief at the time that the genetic map or the genetics of every member of the same species was actually the same. That was the generally accepted belief at the time. Now, Barbara McClintock was a highly respected geneticist, so her results, even though they were scoffed at by many scientists, they were explained by saying that maize is not a natural plant. It's not a naturally occurring plant. It's a crop plant. It's a product of human selection and domestication. So that was generally accepted by people. They could accept that. And this view was actually held right up until the 1960s, when the first transposable element transposable elements were isolated from E. coli, from the fruit fly Drosophila, from yeast, and then from lots of other genomes. Finally, Barbara McClintock's hypotheses were accepted, and she was awarded the Nobel Prize in 1983. All right, but the molecular nature of transposable elements were actually first really understood in bacteria. So we'll start by looking at these. There are two broad types of transposable elements in bacteria, the IS elements and the transposons. That's what they're referred to as. So IS elements can move to new positions in the genome, but they don't carry any genes other than those required for movement. Transposons, on the other hand, also carry other genes. So if we just go back to these IS elements and look at it, look at them down here in a bit more detail. IS stands for insertion sequence. So insertion sequence elements can move to different positions on a chromosome. In the process of this movement, they may interrupt a gene sequence, and interrupting one gene sequence may actually block the expression of other genes that are found in that cluster of genes, which is called an operon in bacteria. So, for example, these IS elements were first identified when they were found to be disrupting an operon in E. coli called the GAL operon. This is basically a cluster of 3 genes sitting in tandem, and they're required for the metabolism of the sugar called galactose. And several E. coli mutants were found that couldn't actually use galactose as a carbohydrate source because of this reason. Okay. All right, so all of these IS elements encode an enzyme called the transposase enzyme. And this is the enzyme which allows the movement of the IS element from one site to another within the genome. All IS elements also begin and end with short inverted repeat sequence that are actually required for their mobility. So even though they eventually found several different types of IS elements in the E. coli genome, they all share these features that are described on this slide. Okay, let's turn our attention to what are called R factors. R factors, or resistance factors, are plasmids. And these are plasmids that carry genes encoding for antibiotic resistance. And this will take you back to one of your pracs, the P-GLOW prac. So let's look at R factors. How do they pick up genes that confer resistance to new antibiotics? They can do this, but how do they do this? And keep in mind that R factors can be very readily transferred to other bacterial cells, or they can be transferred, the R factor genes can be transferred from the plasmid itself, from the R factor plasmid itself, to the chromosome of the bacterium, or from one R factor plasmid to a different R factor plasmid. All right, so how is it that they can pick up new additional antibiotic resistance genes to generate what's called multiple drug resistance? Turns out that antibiotic resistance genes reside on genetic mobile elements called transposons. All right, let's look at some of the structural features of two types of transposons. So at the top here, we have what's called a composite transposon. All right, we have within this composite transposon, and this one is actually called TN10, it contains one antibiotic resistance gene. This encodes resistance to the antibiotic tetracycline. And at either end of the gene, we have this IS10 region, which you can see here. And each of these IS10 regions in this transposon carries a transposase gene, but usually only one is functional. All right, so the IS10 elements that we can see there, as indicated by the arrows, are oriented in reverse directions. That is, these represent the inverted repeats, which are very typical of these transposons. All right, in B, we have an example of what's called a simple transposon. This one is called a TN3, transposon TN3. And it also contains these inverted repeats at either end, indicated by the IR. There are also inverted repeats, that is, they run in reverse directions. But unlike the composite transposons, these IR regions lack the transposase gene. Instead, the transposase gene is here, in between the IR repeats. There it is. So they encode, simple transposons encode their own transposase, and they also contain another gene called the resolvase gene. And this encodes a protein that enables the transposon to recombine very readily into another part of the genome. And of course, where's the antibiotic resistance gene? Here it is in green, and this gene encodes resistance to ampicillin, just like the ampicillin resistance gene that you used in your P-GLOW plasmid. All right, down here we have a plasmid called an R factor, and this R factor, of course, contains several different transposons. It has a TN3, a TN5, and a TN10. And these transposons each carry different antibiotic resistance genes. For example, resistance to kanamycin, ampicillin, sulfonamide. There's one for mercury, resistance to heavy metals, tetracycline, and so on. And these transposons can actually move as clusters of resistance genes. All right, so how do transposable elements move? In other words, what are the mechanisms of transposition? All right, we've already seen that movement depends on the action of a transposase enzyme, which plays a role in both the excision of the transposon and insertion into a new location. All right, most of the transposable elements in both prokaryotes and eukaryotes employ one of two mechanisms of transposition: the replicative pathway and the conservative pathway. In the replicative pathway, a new copy of the element is actually generated, and that copy of the element will appear at the new site, while the original copy, that is the old copy, remains at the old site. In the conservative pathway, the element is actually excised from the chromosome or from the plasmid, and it's integrated into the new site. So this is referred to generally as cut and paste. Okay, that's illustrated here, the two modes of transposition. Here we have the replicative mode here. So let's just note, here we have a TN3 sitting in between A and B, which may be genes A and B. This is going to be the target site, and we've got genes C and D sitting here. So after transposition, using the replicative mechanism, TN3 will actually make a copy of itself so that the original remains after that copy is made, and then the copied version inserts here in between the original C and D in the target site. Okay, so we can call this the copy and paste mechanism. In contrast, in the conservative transposition mechanism, in this example, we've got TN10 in between A and B. Here's our target site with C and D. TN10, rather than copying itself, it's actually excised from that site, leaving A and B behind, and the TN10 then inserts itself in between C and D. So this is referred to, as I said, as cut and paste. That's a really easy way to remember it. Copy and paste and cut and paste. All right, so far what we've been looking at are termed class 2 DNA transposons. Class 1 transposons are very different, very different. They are actually termed retrotransposons. And we need to go back to some early studies that were done in eukaryotes, where they used yeast as a model eukaryotic organism. And these yeast researchers were actually studying gene regulation of this gene here, the His4 gene. And the His4 gene, if you're interested, encodes the amino acid called histidine. And the researchers, they isolated over 1,500 mutations in this gene, in different yeast cells, of course. But two of the mutant lines actually had unstable mutant phenotypes. They were termed pseudorevertants, and this is because these two yeast lines were 1,000 times more likely to revert to wild type than all the other mutants, and that's why they were called pseudorevertants. And it was discovered they looked closer at these two mutants, and it was discovered that these two mutants had large DNA sequences inserted into the His4 gene, but surprisingly, that inserted DNA resembled a class of animal viruses of animals called retroviruses. All right, so to understand what was happening there, we need to look a little bit at the life cycle of a retrovirus. And of course, probably the best known retrovirus is the HIV virus that causes AIDS. Okay, so a retrovirus is let's start at the top here. In looking at this life cycle, a retrovirus is a single-stranded RNA virus. That is, as its genetic material, it carries a strand of RNA. It doesn't carry DNA, just a strand of RNA. That's its genetic material. That RNA is enclosed in a protein coat, which is called a capsid. There's the term there. And that capsid is enclosed in a membrane, which is referred to as an envelope. This particular retrovirus carries its own enzyme. So not all viruses carry enzymes, but this one does, and the enzyme is called reverse transcriptase. All right, the virus in this complete state is actually correctly referred to as a virion. A virion. All right, so when the virion infects a host cell, the entire virion doesn't enter the cell. Just that inner capsid portion will enter the cell. There you see it here. Here's the cell, the host cell, the infected cell's nucleus, and one of its chromosomes is pictured here. All right, so the capsid enters the host cell and leaves its external envelope membrane on the plasma membrane of the infected host cell. Then inside the cell, the capsid will break down, and the enzyme that it was carrying, the reverse transcriptase enzyme, will reverse transcribe the RNA into DNA. So it will carry out reverse transcription. So remember, transcription normally proceeds from DNA to RNA. This is the reverse. So it's called reverse transcription. So here's the original RNA strand that was found in the virion, and here's the complementary DNA that's been synthesized to it using that enzyme reverse transcriptase. Then that RNA molecule will break down. The reverse transcriptase enzyme will synthesize a second strand, complementary strand from that one DNA strand. So now we have double-stranded DNA in the infected host cell. That double-stranded viral DNA is then integrated into the chromosome of the infected host cell. There it is there, and now it's referred to as proviral DNA. Now, when this infected host cell transcribes its own genes on its own chromosome, it will also transcribe the virus genes, the proviral genes. Okay, and this is why we say that viruses tend to hijack the molecular machinery of the cell, the cells that they infect, because they're basically forcing the cell to transcribe the viral genes. Okay, so here we have transcription of the proviral DNA into viral messenger RNA, and now we have all these replicated copies of the original RNA that you would find in the complete virion. Those viral messenger RNA molecules that have been synthesized then can be enclosed within their protein coats to form capsids, and those capsids can exit from the infected cell. When they exit, they'll take little bits of membrane with them from the plasma membrane, for example, and those bits of membrane will form this envelope, and now we have some new virions being formed. So that represents the life cycle of a retrovirus. All right, now we can understand these retrotransposons that were found in the two yeast strains, two mutant yeast strains. All right, so let's think about viral nucleic acids. The retrovirus nucleic acids encode at least 3 different proteins that are required for replication of the virus inside an infected cell. The three proteins, or the three genes that encode those three proteins are referred to as GAG, POL, and another one that's referred to as ENV. ENV. Now, the ENV gene encodes the proteins that go into making that capsid, that protein coat that will enclose the new RNA molecules. In other words, to form new viruses, new virions. And that ENV gene is essential in order for the virions to actually leave the cell, the infected cell, and therefore infect other cells. All right, so in the mutant yeast lines, what they found within this transposable element in the His4 gene, they found two of those three retrovirus genes. They found the GAG and the POL genes. So when the His4 gene with the inserted transposable element in it was transcribed to produce RNA, that RNA could be reverse transcribed into DNA, but the RNA itself, that this retroviral RNA that was found in the yeast gene, actually couldn't leave the cell. This messenger RNA couldn't leave the cell, which they normally would if they were packaged within the ENV protein, because of course within this retrotransposon there was no ENV gene. So instead the DNA copies of that messenger RNA are simply inserted back into the genome of the same yeast cell. And this formed the basis of that instability. The instability and also the ability to revert back to a wild type His4 gene. Oh, okay. So very interesting there. So what this is suggesting is that some transposons, at least, in particular these retrotransposons, actually contain remnants of virus nucleic acids. Absolutely fascinating. All right, DNA transposons for gene discovery, and we are nearly finished. So DNA transposons are quite frequently used as genetic tools, and they're used by geneticists in a variety of different organisms. The mobility of these transposons are exploited to create mutations by insertion, used to tag genes that they would like to clone, and also to insert certain transgenes into the genome. All right, here's a new concept for you, and this one is called the C-value paradox. So the DNA content of an organism, which is generally referred to as the C-value, varies dramatically, as you already know, in eukaryotes, and the amount of DNA doesn't necessarily correlate with the complexity of the organism. So, for example, the genome of the salamander is 20 times bigger than that of the human. The genome of barley is 10 times larger than rice. So no correlation there with complexity of the organism. But a large fraction of eukaryote genomes contain a lot more repetitive DNA than others. And some of this repetitive DNA sequence is very similar to the transposons and retrotransposons that are found in plants. So not just in yeast, but they're found in plants and also in insects. It's thought that almost 50% of the human genome is derived from transposable elements. In other words, we seem to have a lot of remnants of transposable elements or retrotransposable elements existing in our genome. Very briefly, there's two broad types of retrotransposons, which you've probably heard of before. The long interspersed elements called lines, and they represent about 21% of the genome. And there's probably, these are fairly long, up to about 5 kilobases in length, and there's anywhere up to about 40,000 of these, 40,000 copies of these in the genome. And these lines are autonomous elements, so they actually move around, or they can move around like the retrotransposons. Then we have these shorter interspersed elements called signs. So these are only up to about 300 base pairs in length. It's been estimated that there's about 1.5 million signs in our genome, and they represent about 13% of the total genome. And these are non-autonomous elements. And possibly, they're possibly mobilised by the same enzymes that are encoded by and mobilise the lines. So that raises a question. How do plants and animals survive with so many insertions, so many of these transposons and retrotransposons in the genome? So much mobile DNA in the genome. All right, so there's 3 points to consider which can help to answer this question. So firstly, all transposable elements that are inserted into introns, will be spliced out during the processing, normal processing of RNA, because of course introns are spliced out. So in that regard, gene function, normal gene function is retained. But of course, transposable elements would be able to insert at random into either introns or exons. But of course, only those in introns are retained because they're only the ones that are retained are those that don't cause any serious mutations. Because any of them that are retained in the within the gene itself that cause these serious mutations would be subject to negative selection pressures. Okay, so that basically covers that point there. But of course, there are a few active transposons that have escaped from these types of controls. And here's some examples. So haemophilia, the disease known as haemophilia A, is due to 3 transposon insertions in this particular gene here. Haemophilia B is due to insertions in this gene. This disease called neurofibromatosis is due to transposable elements disrupting this gene. And this type of cancer, this type of breast cancer, is due to transposable elements disrupting this gene, which has been given the name BRCA2, which stands for breast cancer 2 gene. All right, and here we have just one example of a human gene. This is the HGO gene, and you can see all those transposable elements that are found in the one gene. All right, that brings us to, I think, our last slide. The last slide of, it is the last slide of applied genomics. All right, so let's look at these grasses here. That have all thought to have evolved from an ancestral grass, a common ancestor about 70 million years ago. All right, barley, rice, sorghum and maize. And since that, in that time, they've all accumulated transposable elements at different levels. So let's check out the large transposon clusters, which are in green. So in green, we have transposon clusters, and we can see that there's an enormous number in barley. And also in maize, lots of transposon clusters in maize compared to sorghum and rice. So the green areas represent the transposon clusters, the orange areas represent genes. So these 4 genomes are actually very similar with respect to the content, gene content, and the organisation of the genes. So the regions, all these different gene regions can be directly compared. In maize and barley, the genes are actually separated, as we can see, by very, very large regions of transposons. All right, and that completes our lectures for applied genomics.
Woo.
Oh, we finished with a big bang, didn't we?
Yes, yeah. Quite interesting, Al.
Low battery.
Sorry?
Good timing, because my battery just came up with a low battery.
Oh, did it? Yeah, I think mine's about on its way out too. Yeah, so there you go. I hope you found that very interesting.
Yes, definitely was.
I certainly do, and you can probably appreciate that there's so much still that's not known.
So I guess the transposable elements, they would be responsible for evolutionary changes, or?
Absolutely.
Yeah.
So if you imagine, you know, all the possible outcomes from transposons, you know, moving to different parts of the genome, moving, not even just moving into genes, but moving into regulatory elements of genes, so that they might, you know, affect how that gene is expressed in a different individual. You know, so we've got the same genes, but, you know, one particular gene that you have might work a lot more efficiently than mine. And that could be a reason to explain that.
Yeah. And I guess viruses too, that's interesting how they can sort of, you know, jump in and change some of the, you know, I guess some of our DNA over, you know.
Absolutely, yeah. And it's fascinating to think that we've got all those remnants, all those viral remnants within our genome, and that they could be having, you know, some impacts as well.
Yeah. Yeah.
Yeah, absolutely. Yeah.
Very fascinating. Yeah.
Very interesting. Very interesting. And not to mention, of course, that the same thing is happening in plants.
Yep. Yeah.
Same thing is happening in plants. You know, so I wouldn't be surprised if, you know, you have, you know, a very sort of homogeneous crop of plants, and then you have one standout, one that's very different from all the others. This could be a possible reason to explain that. Why you have one plant that's, you know, super, has really high vigor, or it seems to withstand, you know, the drought better than the other plants. You know, we don't really know. Like I said, there's so much that's not known about what's going on in the genome. Yeah, really interesting stuff.
Yeah. Oh, I'm sure there'll be other subjects where we can we'll be applying a bit more of this sort of stuff.
Yeah. Yeah, absolutely. So that's it for tonight. That's it for tonight. That's it for the subject. And I'll just stop the recording there.